Hello, everyone. My name is Kannammai Pichappan, and I am very excited to share with you my project on a novel low-cost treatment for PNS neurodegenerative diseases, ascorbic acid. I want to start by highlighting the word low-cost in the title, because in today's healthcare industry, we're all very focused on coming up with high-efficacy drugs and top-notch treatments, which are inevitably expensive. And this snatches it right out of the hands of third world countries and financially struggling people. In America itself, 3 out of 10 adults skip their prescription medications due to the high costs. One of the most expensive diseases to have is a neurodegenerative disease, which are characterized by breakdown of the nerve and are lifelong and cost approximately $84,000 annually to treat, with patients paying more than 30% out of pocket for these drugs, an extreme financial burden. Therefore, my research focused on peripheral nervous system neurodegenerative diseases. The peripheral nervous system controls all motor activities. Therefore, symptoms include difficulty walking, muscle weakness, and muscle spasms in diseases like Charcot-Marie-Tooth disease and Guillain-Barré syndrome. But what happens in a neurodegenerative disease at the cellular level? Well, the elongated blue structure is called a nerve axon, and the nerve impulse travels down the nerve axon. And the purple structure wrapped around the nerve axon is a gel-like substance called myelin, which insulates the nerve signal. When a nerve impulse jumps down a myelinated axon, it jumps from node to node, the sections in between the myelinated segments, in a process called saltador conduction. Since the nerve impulses jump over the myelinated segments of the neuron, an impulse travels much faster along a myelinated neuron than along a non-myelinated neuron. The process of myelin wrapping around the nerve axon is called myelination. However, in demyelination, the myelin becomes damaged, and this is characteristic of neurodegenerative diseases. But where does this myelin come from? Cells called Schwann cells wrap around the nerve axon and produce this myelin. And a critical component of myelin is MAG, myelin-associated glycoprotein, which conducts signals between the nerve axon and the Schwann cell, which are critical in the myelination process. Furthermore, mutations in the MAG gene have been seen in cases of neurodegenerative and demyelinating diseases. The ECM, the extracellular matrix, is a collection of glycoproteins and enzymes which provide chemical and structural support to the cell, and is also critical for myelination. And the main component of the ECM is collagen, a structural protein. And it leads that collagen is also necessary for myelination. This leads me into the objective of my research. I hypothesized that increased collagen synthesis would increase ECM development, leading to healthy myelination, and increased MAG would increase myelin maintenance and Schwann cell neuronal cell signaling, leading to healthy myelination as well. And if only there was one low-cost compound with minimal side effects that could do all of that. And that I hypothesized to be ascorbic acid. And what's very interesting is that patients with neurodegenerative diseases have decreased levels of ascorbic acid in their blood serum. Therefore, this study sought to investigate ascorbic acid's role in PNS myelination, MAG collagen regulation, and Schwann cell neuronal cell signaling. And I tested my hypothesis with a Schwann cell neuronal cell co-culture model. All of my research was conducted in vitro with cell culture flasks, and I co-cultured Schwann cells and neuronal cells, meaning I basically placed Schwann cells and neuronal cells together in a flask. And it was critical that I co-culture because the myelination process includes many important signals between the nerve axon and the Schwann cell. And it was critical that I replicate these signals. And now the exciting part, the effect of ascorbic acid on this co-culture model. Through testing with an ELISA, I saw that ascorbic acid treatment increases myelin, MAG, and collagen significantly in a dose-response fashion. So ascorbic acid is beneficial in a healthy neuronal microenvironment. Fantastic. But the real question is ascorbic acid beneficial prior to disease attacking the neuronal microenvironment? In other words, it's great that it could help a healthy patient, but we want to help a patient who is being attacked by disease. Therefore, I wanted to stimulate the disease. So I utilized a compound called lysolecithin, which comes from egg yolk, to simulate demyelination. And this is often used by researchers for this purpose. So I treated with lysolecithin, and I saw that MAG and myelin significantly decreased in a dose-response fashion. Therefore, I decided to test in a preventative model, meaning I gave my cells ascorbic acid treatment first, the medication first, and then I gave them lysolecithin. I simulated disease attack. Therefore, it was a preventative model. And I saw that ascorbic acid increases myelin, MAG, and collagen in a preventative model in a dose-response fashion through testing with ELISA as well. And to satisfy my curiosity, I wanted to investigate if MAG was a signaling molecule in the myelination process, because the vast majority of intercellular signaling molecules are membrane proteins. And in order to see if MAG is a membrane protein, is located on the cell membrane, I performed an immunostaining for MAG. And the red represents MAG and the blue is the nucleus for localization purposes. And as you can see, what's unique about the staining is the cells were not permeabilized, which basically means I did not poke holes in the cell membrane. So whatever MAG is visible is on the cell membrane. Therefore, this suggests that MAG is a membrane protein and plays a role in Schwann cell neuronal cell signaling in the myelination process. Finally, I wanted to see if the cells were overall healthier and more efficient with ascorbic acid treatment. So I measured something called mitochondrial superoxide, which cells release when under stress through a mitosox assay. Mitochondria produce the superoxide anion as a byproduct of cellular respiration, which in anything more than minute quantities can cause intracellular damage, apoptosis, and even the development of a toxic microenvironment for the cells. And the cells release an excess of this when under stress. However, with ascorbic acid treatment, I saw that superoxide stress signaling significantly decreased, suggesting that the cells are overall healthier and more efficient. Therefore, this research suggests that ascorbic acid treatment increases collagen synthesis, increases MAG, increases myelin, and there decreases stress-related signaling. And this is all fantastic. It's wonderful. But there's a little issue. This is all in a preventative model. And neurodegenerative diseases are very difficult to diagnose early on, let alone before the disease even attacks. Therefore, I developed a disease prediction regression model, which means that I found a correlation between myelin and other biomarkers in the body, such as collagen, ATP, ascorbic acid, etc., that can be non-invasively, safely, and accurately measured in the blood serum or in the body. And I trained this model with my own cell culture data, and after testing this model, I saw that I had 85% accuracy, which is the gold standard when it comes to disease prediction. And as you can see, the orange are the values that the model predicted, and the red are the actual values, and they're not too far apart. And such disease prediction regression models have the ability to revolutionize the healthcare industry and patient diagnosis, as this model can predict susceptibility to a neurodegenerative disease, but similar models can be developed for a variety of other diseases as well. In conclusion, I don't plan on stopping here. I want to develop disease prediction models for other diseases as well, investigate ascorbic acid's role in the CNS, and look into in vivo studies, because a major limitation of the study is that all of this research was conducted in vitro in a plastic flask. And this is under the assumption that everything works the same way in a plastic flask as it does in the human body, which is not at all true. Talking about applications of this research, incorporation of ascorbic acid into pharmaceutical drugs could significantly lower the price and minimize side effects, especially if it is used as a combinatorial treatment with existing prescription medications, as it could lower the quantities of prescription medication that is required for treatment. Currently, there is no use of ascorbic acid in medications for PNS neurodegenerative diseases. However, ascorbic acid has been safely administered in clinical trials for other purposes, suggesting that it is biocompatible and safe for human use. Incorporating ascorbic acid into PNS neurodegenerative treatment regimens could significantly lower the price, and hopefully this inspires the development of other low-cost treatments in an effort to increase accessibility of medication to the patients. Of course, I would like to thank Ms. Leonardi for being a fantastic research mentor through my favorite experience of high school, the Bergen County Academies for generous funding with lab equipment, and NJAS for providing this fantastic opportunity. Thank you.